Good morning, everybody. Is this on? I'm Jennifer Fangaskin. I'm from Melbourne. I've got great pleasure this morning in chairing with Professor Stephanie Watson the Australian Vision Research Plenary session. So this morning we've got 5 great papers to showcase to you some of the research that AVR is funding. Because of interest for time, can I ask everyone to keep questions until the very end of the session? And I'd like to introduce our first speaker, who's Dr. Vivek Gupta from Macquarie University, and he's going to be speaking on tau targeting to protect retinal ganglion cells and the optic nerve in glaucoma. Now, Vivek's pre-recorded his talk, but I believe, oh, you're here. And he'll be here to answer any questions at the end. Thank you, Vivek.
Take it away.
Hi, I am Vivek Gupta from Macquarie University, and today I'm going to
microphone down a bit towards you.
And today I'm going to talk about the tau targeting to protect the retinal ganglion cells and the optic nerve in glaucoma. So tau protein is extensively expressed in the central nervous system, and it plays a key role in preserving the cytoskeletal architecture and its stability in the axons. So we know tau protein that it's a key mediator of neurotoxicity in diseases like Alzheimer's disease and frontotemporal dementia and several other tauopathies. But recent research also shows that it plays a role in glaucoma pathogenesis. And here in the human postmortem tissues, we show that there's increased tau phosphorylation in the inner retina, and the findings are also supported by similar results in the Western blotting, where several-fold increased tau phosphorylation was observed in the human postmortem samples from glaucoma patients. Now, coming to the glaucoma animal models, there's a research to support that the tau co-expresses with the TUJ1 marker, which is a marker of the retinal ganglion cells. And our studies have recapitulated these findings, and we have observed the increased tau hyperphosphorylation in the inner retina in the ganglion cell layer. So based on these findings, we asked ourselves the question whether tau upregulation can exacerbate the inner retinal damage in glaucoma, and conversely, if we knock down tau expression in the ganglion cells, can we protect the retina? So for this, we synthesized the AAV9 constructs, and these constructs were to overexpress the tau protein or to knock down the tau protein. And these were administered to the animals through the tail vein injections. And here towards the right, you can see the green fluorescent protein expression in the fundus images. So there's a comparable expression in the overexpression as well as in the knockdown groups. The expression of tau protein was also confirmed in the retinal sections. So in the top panel, you can see that there's a GFP expression in the overexpression group and in the knockdown groups. In the middle panel, there's a tau protein expression, which is increased in the overexpression groups, but several-fold decreased in the knockdown. And in the bottom panel, you have the phosphorylated tau. And the findings were also confirmed in the Western blotting. We observed approximately a 6.5-fold upregulation of tau protein in the overexpression group, and approximately a 5-fold downregulation of the tau protein in the knockdown group. The effects of tau modulation were then confirmed using the electrophysiological recordings. The electrophysiological recording that we used was positive scotopic threshold response. It reflects the inner retinal function. And as you can see, that under the normal physiological conditions, whether we upregulate the tau protein or downregulate it, it has a detrimental effect on the retinal function. And similar observations were seen in the retinal histological examination, where the ganglion cell layer density was significantly decreased in the overexpression as well as in the knockdown model. So these studies show that tau protein plays an important role in the maintenance of retinal function and structure under normal healthy conditions. Now, what happens in the glaucoma conditions? We subjected the animals to increased IOP using the microbead injection. So this experimental paradigm increases the intraocular pressure from, say, 10 millimeter of mercury to around 25 to 30 millimeter of mercury. And as you can see in the pink trace in the bottom, the group that had the glaucoma and also had the tau overexpression showed significant decline, a massive decline in the scotopic threshold response amplitudes. The retinas were also examined using hematoxylin and eosin staining, and here we observe a decline in the ganglion cell layer density in the glaucoma, but more decline, further decline in the groups that had glaucoma and also the tau overexpression. Now, what happens if we subject the animals to AAV9-induced tau suppression, tau knockdown? So in this case, interestingly, we observed that the glaucoma group showed a decline in the positive STR amplitudes, but with the tau knockdown, there was a partial rescue of the scotopic threshold response amplitudes. Similar findings were observed in the histological analysis, where there was a protection against the ganglion cell layer density loss in the group that had the tau knockdown. So based on this, we can say that there is an increased expression of tau in the glaucoma, and if we increase the tau expression further, it has a detrimental impact. But if we knock down the tau, there is a protection of the retinal structure and function. But the challenge is to regulate the ideal levels of tau protein to make it meaningful clinically. We also examined various changes that occur in the biochemical milieu in the retina in response to tau modulation, and we observed that the phosphorylated neurofilament heavy chain protein, which is a marker of the neuronal integrity, its levels were decreased in glaucoma in the optic nerve. They were further decreased in the group that had the tau overexpression, but there was a rescue observed in the group that had the tau knockdown. Now, going further, we examined the effects on tubulin. Tau protein interacts with the tubulin and maintains the cytoskeletal integrity. The acetylated tubulin is more stable form of tubulin. And here in this immunostaining, we see that acetylated tubulin was decreased in glaucoma. It was also decreased in the tau overexpression paradigm, but its levels were increased in the tau knockdown group. Microglial activation has been shown to occur in several neurodegenerative conditions in the brain and also in the retina. And we observed that there was an increase of the microglial marker IBA1 in glaucoma, a further increase in the overexpression group, but a significant decrease in the IBA1 expression in the group that had the tau knockdown. And we observed these findings in both the optic nerve and the retina of the animal group. So finally, we examined the changes that occur in the apoptotic changes that occur in the retina in response to tau modulation, and we used the tunnel staining as well as the cleaved caspase 3 immunostaining. And in both of these experiments, we observed that the tau knockdown gave a significant protection to the apoptotic processes against the apoptotic processes in the retina in the tau knockdown group. So, concluding, we can say that tau is essential under the normal conditions. It is a very important protein, but it undergoes hyperphosphorylation in glaucoma conditions. So if we knock down tau protein, it can provide structural and functional protection, and also it can rescue the biochemical environment in the retina. These findings taken together provide a strong incentive to further explore tau as a retinal biomarker for detection and drug targeting in glaucoma. Of course, to determine how to regulate the tau levels to the appropriately is going to be a challenge. So I would like to thank our group, Professor Stuart Graham, and also towards the right we can see Dr. Kanishka Pushpita, who did a major part of the work here. Thank you very much.
Thank you. So we'll have questions at the end. I'd now like to introduce Associate Professor Fan Zhu from the Sydney Pharmacy School, where she's Head of Molecular Drug Development Unit. She's been working with Professor Max Conway. In the presentation, we have on developing novel therapy for human uveal melanoma. Is this recorded? It's a recorded presentation.
At the Annual Congress of RAMSCO today, my topic is developing novel therapy for human uveal melanoma. First, I'd love to acknowledge the traditional owners of the land. I would like to pay my respect to the elders, past, present, and emerging. Cutaneous melanoma is well known to us, especially Australians, but uveal melanoma, or so-called is not very well known to most people, because it is a rare type of cancer. is the most common primary intraocular malignancy in adults, and the second most common melanoma. Its global incidence is about 8 cases per million. It arises from uveal tract. Although rare, is very deadly. It is a highly metastatic disease, with up to 50% patients develop metastatic disease, leading to death within 6 to 12 months. The overall survival of is very poor. The median survival is about 5.2 months in metastatic patients. In clinical setting, the local treatment for primary mainly includes laser treatment, brachytherapy, and enucleation. Radiotherapy and laser treatment are more effective for small tumors. These local non-pharmacological therapies are effective for patients with early diagnosis, but in general, result in unsatisfactory outcomes such as impaired vision, blindness, and impacted quality of life. On the other hand, there are very limited pharmacological therapies available for at present because its pathogenesis is poorly understood. Enormous efforts have been made to explore new drugs for Cytotoxic drugs, targeted therapy, and immunotherapy are the main categories of agents under investigation in laboratory or clinical trials. Most of the agents tested have showed unsatisfactory clinical outcomes. However, it is encouraging that tebentafusp has been approved to treat a small population of patients expressing a specific type of human leukocyte antigen. Nevertheless, it is highly desired that novel drug candidates can be identified to better treat primary and metastatic Therefore, our team has been dedicated to develop novel therapies for with high efficacy and manageable toxicity. Generally speaking, drug development pipeline involves preclinical and clinical studies. There are two main approaches for target identification. Because we know little about the pathogenesis of we adopt physiological-based drug discovery approach. It is to screen our candidate molecules based on readouts. We have widely screened natural compound libraries, existing drug pools, as well as chemically designed and synthesized novel molecules in-house. Upon identifying our lead molecules, we then move on to a series of preclinical characterization, such as in vitro evaluation, as well as ex vivo and in vivo validation. In today's talk, I'm going to show some of the preclinical data we have collected for one of our lead molecules identified in-house. Mitochondria are the key organelles in cells, which function as the powerhouse of the cell. Mitochondria control cell survival, cell death, and cell cycle. They mediate fatty acid synthesis and metabolism, as well as have many other physiological functions. Research showed that mitochondrial function differs a lot between tumor and normal cells. They are essential for the survival and the reproduction of cancer cells, so mitochondria have been considered as the most important anti-cancer target. Next, I'd like to talk a bit about fatty acids. They play key roles in human body. They are the building blocks of the fat in our bodies and in the food we eat. There are saturated, monounsaturated, and polyunsaturated fatty acids naturally occurring in our diet. It is known that some polyunsaturated fatty acids, so-called PUFAs, have anti-cancer property, such as omega-3 fatty acids. Some of the PUFA metabolites may also have anti-cancer effect, like epoxy fatty acids, but the naturally occurring epoxy fatty acids are not suitable to be anti-cancer drugs. Therefore, chemically synthesized epoxy fatty acids have been widely studied as potential anti-cancer agents. In this study, we designed and synthesized a group of fatty acid derivatives to be evaluated in our study for anti-UM property. Here I'm going to focus on our findings regarding a lead molecule, which was named as novel molecule 2 NM2. Because these molecules are under IP protection, I'm unable to disclose their chemical information here. We have used a range of molecular methods in our drug evaluation study. First, we assessed the anti-cancer property of NM2 in immortalized cell lines, as well as primary cells derived from patient tumors. The cell lines used in our study include C918, 92.1, and MP46 that are primary tumor-derived cell lines, as well as OMM1, which is a commonly used metastatic cell line. I also want to flag that we include normal ocular cell lines in our study to assess the tumor selectivity of our compounds. More importantly, the primary cultured cells derived from patient tumors are the unique resource available in our study. To study the anti-cancer effect of molecules, we often use colorimetric assays. Some of them have been listed on this slide. We assess the viability of cells with or without NM2 treatment using MTT and alarmin blue assays. Both assays are classic methods established for anti-cancer drug studies. We also use the LDH assay to evaluate NM2-induced cytotoxicity. And as mentioned before, NM2 is a fatty acid derivative, which highly likely impacts on mitochondria. So we also included two commonly used mitochondrial functional assays in our study. ROS assay reflects the oxidative status of cells, while JC1 assay indicates the membrane potential of mitochondria. Our data show that 2 micromole NM2 treatment for 24 hours can potently reduce the cell viability in all 4 cell lines as to both assays. Consistent finding was also obtained in our confirmatory experiment based on 4 patient tumor-derived cell lines. On the other hand, there is mild to moderate cell viability reduction observed in non-tumor ocular cell lines, which indicated that NM2 has favorable tumor selectivity. In our further in vitro study, we derived the IC50 values of NM2 in the 4 immortalized cell lines. IC50 value is the compound concentration required to reduce cell viability to half of the maximal effect. As shown on the slide, the IC50 values of NM2 range from 0.17 to 1.27 micromole in the 4 cell lines. This finding tells us that the anti-cancer property of NM2 is very potent, which is much higher than most of the drug molecules reported in literature and clinical trials against the In the cytotoxicity and mitochondrial functional analysis, we showed that NM2 can significantly induce the cell death across all 4 cell lines. Such cytotoxic effect is accompanied by increased oxidative stress and impaired mitochondrial membrane potential. Our further study assessed the cell death profile of cell lines treated with or without NM2 using annexin 5 PI staining flow cytometry. This slide shows the representative image obtained in MP46 cell line. We found that NM2 mainly induces cell necrosis rather than apoptosis in MP46 cells. Consistent observation was also made in other cell lines as well as the cells derived from patient tumors. Necrosis is a type of uncontrolled, irreversible cell death mechanism that results in swelling of the cell organelles, plasma membrane rupture, and eventual lysis of the cell. the spillage of intracellular contents into the surrounding tissue, leading to tissue damage. In the recent decade, such cell death mechanism has attracted a lot of attention in anti-cancer drug research. As mentioned earlier, the high metastatic rate is the major cause of mortality in We then assessed the anti-metastatic effect of NM2 in Here I'm going to show our data obtained using wound healing assay and colony formation assay. Wound healing assay studies the inhibitory effect of compounds in cell migration, while colony formation assay evaluates the impact of compounds on cell reproductive growth. It is known that cell migration, invasion, and reproductive growth are the key events involved in cancer metastasis. My team is currently working on data collection using cell invasion assay as well. We found that NM2 can significantly inhibit cell migration in all cell lines tested in our study. This slide only shows the representative images obtained in 92.1 cells. Briefly, we made a scratch at 0 hour and then treated the cells with or without NM2 for 24 hours. In the control group, cells migrated rapidly and the scratch fully healed the next day. But in the NM2 treated group, the cell migration rate is significantly reduced and the scratch did not fully heal after 24 hours. In the colony formation assay, we first treated the cells with or without NM2 overnight. We then subcultured the cells and maintained for 8 days. We observed that in the control group, cells propagate normally, while in the NM2 treated group, there were much reduced number of cells survived and propagated. This data suggested that NM2 potently suppresses reproductive cell growth in cells. Overall, we can see that NM2 is a potent anti-UM candidate drug. It remarkably reduces cell viability, significantly impacts all mitochondrial function, and induces cell necrosis in cell lines. It also shows potent anti-metastatic effect. Our future work will explore its cell-killing mechanism, identify its molecular targets, as well as investigate its in vivo effect using our established xenograft models, as well as small animal PET imaging. At the end of my talk, I'd like to acknowledge our students and collaborators who greatly contributed to this project, especially the Ramsco Fellows, Associate Professor Max Conway and Dr. Lian Lim, as well as Professor Svetlana Cherepanov and Associate Professor Michelle Madigan. Their continuous input made our project moving smoothly. I also want to thank Australian Vision Research and AUREA for generous funding support. Here is the reference list. I'm happy to take any questions from the audience. Because I'm unable to attend in person, please also feel free to email me any questions you have later on. I will communicate with you via email exchange as well.
Thank you, Fan. That was a fascinating presentation. We'll move on to Associate Professor Peter van Wijngaarden, who's an ophthalmologist from Melbourne and Deputy Director of the Centre for Eye Research Australia. Over to you, Peter.
Thank you very much. It's my pleasure to be talking today about non-invasive detection of retinal ischemia using hyperspectral imaging. So why ischemia? Well, we know it's a driver of many retinal diseases, and identifying ischemia is really important for stratifying risk for our patients, and particularly for neovascularization and macular edema, sight-threatening sequelae of ischemia. Now, we've got excellent tools to identify people at risk or with ischemia in the clinic, but they have their limitations. We know fluorescein angiography is the gold standard, but it's invasive, it's time-consuming, so it disrupts clinical flow. It's costly and it carries some risk to our patients. OCTA, it's an excellent modality. It's non-invasive, high resolution, and gives us depth-resolved imaging of the circulation. But the lower cost machines that most of us can actually afford provide us with suboptimal resolution. They're cumbersome to use at the moment, and long acquisition times are often difficult for our patients. And again, most of the commonly used devices have quite narrow fields of view, so it's hard to see more peripheral ischemia. So we have been working with a technology known as hyperspectral imaging, and to understand that technology, it's worthwhile just drawing a brief comparison with conventional retinal photography. So when we take a photograph of the retina using a single white light flash, we get a colour image that recapitulates what we see as clinicians during examination. That's great for clinical recording, but it's actually a fairly poor data source. Essentially, it's just got 3 channels of information. In contrast, in hyperspectral imaging, we acquire a rapid series of images of the retina with different wavelengths of light, and that generates a cube of data. It's a really rich data source, and when we combine that with machine learning algorithms, we can detect biomarkers of diseases that we can't see with our naked eye. So with a hyperspectral camera, the camera sensor measures light intensity at each location at each wavelength. And the underlying premise is that the physical interaction of light with the retina is dependent on the structure of the retina. And so reflected light is captured by the camera sensor and provides us with very useful information about that structure. Now, as the interaction between light and the retina varies as a function of wavelength, imaging an object sequentially with different wavelengths of light provides us with much richer information about the retina than is gathered with a single white light flash. So our team's been working with this technology for the last few years, and we were the first group in the world to successfully apply this technology to detecting amyloid beta in the retina. It's a protein that accumulates in both the retina and brain in Alzheimer's disease. But specifically in relation to ischemia, the question that we sought to answer with this research is, can hyperspectral imaging be used to detect retinal ischemia on the basis of changes in the reflectance signal of blood within the retinal tissue? And we all know that arterial blood has got a very different colour to venous blood, and the graph shows us there, if you take oxygenated and deoxygenated haemoglobin and look at the absorption profile across different wavelengths, you can see that oxyhemoglobin absorbs very poorly in the red wavelength, and so it appears red, it reflects the red light, whereas deoxy in the blue curve is more absorbent of red wavelengths. And so that's obviously the underlying principle by which pulse oximetry works. Infrared and red light are shone through the tip of the finger. There's a photodetector that can then detect the amount that's absorbed by oxy and deoxyhemoglobin, and that can be inferred by the ratio of absorbance between red and infrared light. And the arterial saturation is gleaned by pulsatile fluctuation, so you can attribute the saturation to arterial blood. So a similar principle is deployed in retinal oximetry. You may have heard of the OxyMap device. This is where the retina is illuminated with 2 different wavelengths of light, and the light reflected from and adjacent to the larger retinal vessels can be used to calculate an optical density measurement which correlates with oxygen saturation, very much like we do in pulse oximetry. It's a very useful technique, but it's limited to larger order vessels because of signal-to-noise challenges. There's a lot of other stuff in the eye that absorbs and reflects light. So we hypothesised that if we have a hyperspectral camera, we could potentially tackle this signal-to-noise challenge by imaging across many different wavelengths of light. And in a pilot experiment, we took 5 healthy participants, we imaged them on a hyperspectral camera, and then we sampled the reflectance profiles in second-order arterioles and venules. And we plotted the average results there for arterioles in red and venules in blue, and you can see that we can tell a difference between arterioles and venules. We did some mathematics using linear combinations of the known reflectance properties of oxyhemoglobin, deoxyhemoglobin, macular pigment and melanin. They're the most absorbent components of the retina, and we found that we got excellent mapping of the actual measured results. And that allowed us to conclude that most of the difference that we're seeing there in the reflectance properties of arterioles and venules is attributable to oxyhemoglobin, supporting the underlying premise. So with that in hand, we embarked on a pilot study. We recruited participants from the laser and angiography clinics at the AVR Hospital in Victoria. We recruited adults who were undergoing fluorescein angiography in the clinic for presumed retinal vascular disease. They had to have had their angiogram within 3 months of hyperspectral imaging and couldn't have been treated for their disease in the interim. And so we defined cases on the basis of having ischemia on their fluorescein angiography and controls as having no identifiable ischemia. We had, by and large, good balance between the groups for demographic factors, age and gender, and you could see that half of our cases had proliferative diabetic retinopathy, a third non-proliferative retinopathy, and 13% with branch retinal vein occlusion and 9% with retinal vasculitis. Our patients were imaged on a hyperspectral camera that can image the retina across 90 different wavelengths of light in less than a second. Unfortunately, that device is quite a narrow field of view, about 25 degrees, so we had to montage these images, and this is just a representation. You've got to think that each of these images is actually a stack of 90 images. So we did that manually initially, but we developed a machine learning algorithm to automate that. We then co-registered that individual's hyperspectral images with their fluorescein angiogram and cropped the angiogram to the same area. We then annotated areas of ischemia on the angiogram images. Again, that was initially done manually, but we've now developed a machine learning tool to facilitate and streamline that. So that was then serving as the ground truth for ischemia for that individual. And we then took the hyperspectral images and said, can we predict in that individual, using machine learning, which of those pixels in the hyperspectral image are actually ischemic, and using those angiogram images as our ground truth. So we developed a convolutional neural network, which is optimised for classification. And essentially it was a binary output. So for each location in the retina or each pixel on the image, it was a simple question, is this ischemic? Yes, no. We had 55 image sets. We took 45 of those for training and optimising the network, and then we tested it on 10 image sets that hadn't seen the neural network previously. These are the results of this pilot study. We showed that at a pixel level, the classification accuracy for the training set was 90.8%, and for the test set, 88.2%, with an area under the curve of 0.93 and 0.86 respectively. And what I'll impress upon you is that that's at a pixel level. So at the individual level, the area under the curve is 1. So it's perfect at detecting ischemia at the individual level. Here's some examples of some of the cases. So you have the angiogram images across the top, the annotations in the next row, and here you see the hyperspectral predictions of ischemic areas. So these are wherever you see a white point, that's where the model is predicting ischemia, and you can see very good correlation between the hyperspectral and the marked angiogram images. We're now embarked on a validation study, so we're seeking to recruit an additional 120 participants with retinal ischemia, and we've made reasonable progress in our recruitment despite the pandemic. The major innovations of this validation study is that we're using a non-mydriatic hyperspectral retinal camera that our group has developed and is in the process of commercialising. We have, that camera acquires 30 images of the retina with different wavelengths of light in a quarter of a second. It has a wider field of view at 36 degrees and higher spatial resolution than the Optina camera. And our ultimate goal is that we can opportunistically detect ischemia in the context of routine photography. This should be the same sort of price and the same clinical workflow as a standard retinal camera. So with that, I'd like to acknowledge the contributors to this research, most notably Xavier Hadou and Davi Dang, the teams at the AVR Hospital and CIRA, and importantly our funders, and I'm tremendously grateful for Australian Vision Research. This is a research organisation by ophthalmologists for ophthalmologists. Thank you.
Thank you, Peter. Fantastic work. I'd now like to introduce Professor Catherine Burden, who is an ocular genetics expert at the Menzies Institute for Medical Research in Tasmania, and she's been working with Nitin Verman and Brendan Vogt. This is a recorded presentation.
Thank you to Australian Vision Research for the invitation to speak in this session today. I'm going to tell you about our project looking at the genetics of treatment response in diabetic macular edema. This work is predominantly that of Dr. Raja Gurung, who is a PhD student in my laboratory, who is supported by a scholarship from Diabetes Tasmania. We're talking about diabetic macular edema, which is the accumulation of fluid in the retina around the macula in diabetic individuals. It is one of the most common causes of vision loss amongst diabetes patients. It is treated with the intraocular injection of anti-VEGF antibodies. So VEGF is an important molecule in the vasculature of the eye, and these drugs block its effect, prevent the leaky blood vessels. They do work remarkably well. Many patients will stabilise their disease. Most will restore their vision, their lost vision. However, they are expensive, they are invasive, with the intraocular injections required monthly, taking a lot of time, and it doesn't work for everyone. The trials show around 30% of patients had unresolved edema after 12 months of treatment, and more recent real-world data backs that up with about a third of patients not benefiting. The treatment also leads to anxiety about the injection, to guilt at the assistance that patients require, and concern at the high cost of the treatment. So it would be great to be able to avoid some of these things, particularly for patients where the drugs aren't going to work. So we would like to have a crystal ball where we could look into and figure out before we start treating someone if they're going to benefit from this or not, and if not, push them towards a different kind of therapy that might stabilise their disease sooner. So we've been working on the diabetic macular edema treatment response study, which is a collaboration with my colleagues at Flinders University. So the participants come from Flinders Medical Centre, the Royal Hobart Hospital and private clinics throughout Tasmania. All the participants, of course, have diabetes and are receiving intravitreal anti-VEGF for the treatment of diabetic macular edema, and they needed to have 12 months of follow-up data available. So we collected a range of data from the medical record, the demographics, the medical history, biochemistry profiles, and of course measures of visual acuity, central macular thickness, and their treatment regimes. And we have visual acuity and central macular thickness at every injection date. So we also collected DNA from whole blood or saliva and have done genome-wide SNP array genotyping and whole exome sequencing. So our outcome measures are the change in best corrected visual acuity after 12 months of treatment, and also the change in central macular thickness over the same time period. We have 220 participants in our cohort where we have complete clinical information at all the necessary time points, as well as DNA information available. So we're looking at both the functional and the anatomical response. They do track reasonably well, but they don't correlate perfectly. So we're looking at both of them. So this is a genome-wide association scan for the change in best corrected visual acuity over 12 months. The x-axis has the position in the genome with the chromosomes laid end to end, and the y-axis has the statistical significance, and each dot is a genetic variant or a SNP that we've assessed. We're looking for the SNPs that make it above the red line, which is our threshold for genome-wide statistical significance, accounting for the number of tests that we've done. So in this scan for best corrected visual acuity, we have 3 loci that meet those criteria. I've listed the gene that is closest to the associated SNP, but these SNPs aren't in those genes. They're upstream or downstream from them. You can see in this table that all 3 loci, the top-ranked SNPs are relatively rare. They have a frequency of 2 or 3% in our cohort, which we were actually expecting to see more frequent SNPs come up. So this is an interesting finding. But even though they're rare, they're having a big effect. So this SNP reduces the visual acuity by 21.8 letters for every copy of the rare allele, which is quite a substantial drop in vision. So this result needs replication in another cohort or in a larger cohort, and we have a similar set of results for the change in central macular thickness as well, and we found 2 loci in that study. So there are some limitations with genome-wide association analysis using these SNP array technologies. If we think of the genome looks like this. It has genes that have exons which code for protein, and the exons are separated by non-coding introns, and there are also non-coding sequence between the genes. So if the genome is represented with a solid line, a SNP array that we used for the genome-wide association looks like this. We're looking at genetic variants that are scattered throughout the genome, represented by each blue line. So some of them are in the genes themselves, some of them are in introns, and most of them are in between genes. So we wanted to start to think about what's actually going on in the genes themselves, and for a specific set of genes that could be important in the response to VEGF therapies. What are the changes in the genes that are important? So funded by the Australian Vision Research Grant, we are doing exome sequencing, which is where we literally sequence all of the exons in the genome and look at what's actually going on in the genes themselves. Now, this pilot project, we're looking just at a subset of our cohort. So we chose the people with the best response and the people with the worst response to compare. And we defined the responders as those with at least a 5-letter improvement or a 10% reduction in central macular thickness in their better responding eye. And the non-responders are those who don't meet either of those definitions. We have 46 non-responders and 109 responders in this pilot project. So to reduce our multiple testing burden in this small cohort, we've chosen a set of candidate genes that we hypothesise might be involved. We've got genes from the VEGF pathway, which is the pathway that the drugs are acting on. We've chosen some genes that have been reported to be associated with treatment response for age-related macular degeneration, which is treated with the same spectrum of drugs. And we've put in the genes that are closest to our association signals in the genome-wide association scans. So these are the preliminary results. These are the top-ranked SNPs from a very quick and dirty association analysis. We've only had these data a few weeks. We have a lot more work to do delving into them. But these 2 SNPs are in the FLT1 gene, which codes for the VEGF receptor. The p-values are at about 10 to the minus 5. They're just shy of statistical significance for the number of tests we did. And they're both actually in the intron. So while we sequenced the exons, along with that we capture a little bit of the intron as well, and both these variants are actually in the nearby intron. And they're both much more common than what we saw with our associated SNPs in the genome-wide association study, but they're very rare in the non-responders. So in this situation, the minor allele here is more likely to make you a responder, to do well on these drugs. These 2 loci came from the genome scans, so they're the genes that are closest to our association signals from visual acuity and central macular thickness. And again, both the variants are intronic. They're both fairly common but much reduced in the non-responders, and they have quite big effect sizes as well. This gene, TBC1D32, we don't know how it's related to diabetic macular edema or the response to treatment. But interestingly, when I was looking through this data, we do see a stretch of SNPs within the gene itself that are not present in the responders. They're only present in the non-responders. So they don't reach any level of statistical significance on an individual SNP basis, but we need to go in and look at that much more closely for a haplotype effect or individual variants that are actually in the exons themselves. So as I said, this is very preliminary work, and we will continue to delve into these data over the coming months. But we're quite excited that between both these data sets we'll be able to find some variants that predict treatment outcomes in diabetic macular edema. So thank you very much for listening. This is my list of collaborators and acknowledgements.
Thank you very much, Catherine. The next speaker is my co-chair, Professor Stephanie Watson. Stephanie leads the corneal unit at Eye Hospital and also heads the research group at the University of Sydney. Thank you, Stephanie.
Thank you. So I'm going to be talking on the bacterial ocular surveillance system. Now, corneal blindness is still a big problem. It affects all ages and it's typically irreversible. So much so that WHO has recognised corneal scarring as a priority eye disease. Now, a key cause is microbial keratitis. And the problem is that for the elderly, 1 in 10 still lose an eye. And children face a lifelong visual impairment or blindness due to amblyopia. Now, this has been recognised to be a big problem, and recently there's been a call for microbial keratitis to be recognised as a neglected tropical disease with the WHO, as this would give the necessary resources behind it to try to really make some impacts in reducing vision loss from the disease. Now, one of the problems in managing microbial keratitis is that there are many bugs that can cause it. And when we had a look in 2012, we found 232 different genera. We repeated this study in 2019, and we found an increase of 456 pathogens with close to 700 different species reported as causing keratitis. And in these species, the fungi had the greatest variety that will cause all organisms. So look, how do we go about improving outcomes in keratitis? Well, there's a number of things we need to do, but a starting point is really identifying the microbe so we can choose the best treatment, and then educating the patients and the public on risk factors for the disease and improving the patient experience. But just back to the bugs and identifying them and how we approach this. So in clinical practice, we begin by documenting baseline indices, performing a corneal scrape, choosing our therapy, which we have to do empirically because it takes time to get our results. We then initiate sterilisation, modify the antimicrobial if needed, and determine our endpoints. And the typical baseline indices that you should look for is thinning, an epithelial defect, infiltrate, and hypopyon, and all of these can be measured. So the starting point to get our bugs is really the corneal scrape. And we looked at the evidence around corneal scraping to find how best to perform the procedure. And these are the steps that should be done, and this work was done with Associate Professor Shameen Samarakrama. And this is pre-COVID, and one of the things we said was wear a mask, which is not so much of a problem these days because we're wearing them most of the time. And as a result, we produced a video and then studied it and showed that the residents and trainees that have watched the video on how best to perform a corneal scrape actually had a higher yield. But antimicrobial resistance, is emerging as a big problem. So much so that there's a global action plan from the WHO on it. And in Australia, bacteria are the leading cause. But our results to find out what the bug is causing the keratitis still typically take at least 48 hours, so our treatment remains empiric, with the current guidelines recommending ciprofloxacin or ofloxacin or fortified therapy with cefazolin plus gentamicin. But look, we wanted to know do these guidelines really reflect the microbes that we're seeing in everyday clinical practice? And so we set up initially the CASP, which was Keratitis Antimicrobial Resistance Surveillance Program, to have a look and find out what was going on locally, and this was first done at the Sydney Eye Hospital. We've then moved forward to produce the BOSS, which is the Bacterial Ocular Surveillance System, and our initial reports from the CASP showed that most of the bugs that we were seeing in 2016 were gram-positives. That's over 83% from keratitis, with most coagulase negative staph. And there was 10% resistant rate to cefalothin, which was a bit of a worry, and 4% of staph resistant to chloramphenicol. Now, gram-negatives composed around 17%, and Pseudomonas was the most common. And in this initial data, there was a little bit of resistance to ciprofloxacin. So this meant that our main the main antimicrobials for our attack against microbial keratitis, based on these patterns, should be gentamicin and cefalothin, or ciprofloxacin or ofloxacin. So what we're doing now with funding from the AVR is establishing a national surveillance program for bacterial ocular surveillance. Corneal scrapes will be performed with local protocols, and a positive culture considered any bacterial or growth present on the solid media. We're going to identify the bacteria with Malditoff, and standard methods for determining antibiotic resistance and sensibility as susceptibilities will be used. And with this, our initial data, and this is data from across Sydney, we had 75% gram-positives, 46% coagulase negative staph from this, and worryingly, resistance to cefalothin had increased to 19%. And this is very high and something of concern. We also noted increasing resistance of MRSA to ciprofloxacin and gentamicin. A quarter were gram-negatives, Pseudomonas the most common, and there was no resistance that we detected in our samples. So based on this, similar cover, our data suggests that similar cover can be achieved with ciprofloxacin or ofloxacin and cefalothin and cefazolin plus gentamicin, but you actually get improved cover if you use chloramphenicol and an anti-pseudomonal agent. So chloramphenicol plus gentamicin was superior to monotherapy with ciprofloxacin or ofloxacin, and was superior to cefazolin plus gentamicin, and this was significant. And chloramphenicol plus ciprofloxacin was superior to monotherapy and superior to cefazolin plus gentamicin. Equal cover was found with chloramphenicol plus gentamicin and chloramphenicol plus ciprofloxacin and ofloxacin. So we have some new tools in our fight against keratitis, and we may want to consider whether cefalothin is still the optimal agent to use with our anti-pseudomonal drugs. It's also important to note that fortified antibiotics versus fluoroquinolones have different profiles in terms of safety. So there's an increased risk of chemical conjunctivitis with fortified antibiotics, ocular discomfort increases, and some studies have reported delayed epithelialization. And the classic signs of toxicity is when you have ulceration and staining of the inferior conjunctiva, and the patient looks down and the superior conjunctiva is white compared to red inferiorly. We also found in our 5-year study of keratitis that patients with ocular surface disease and microbial keratitis, if they were given fortified antibiotics, they actually had a higher risk of complications, a higher need for surgery and longer healing times. So once we've got our empiric data, our empiric therapy started, we then continue this typically for 2 days, day and night hourly, and then assess after 3 days, after 5 days of treatment. The antimicrobials can be modified at 48 hours. So the BOSS data will help us choose our initial treatment, but then we still need to wait to receive the results of antimicrobial testing and alter if necessary. Determine your endpoint. You need to know generally by about 5 days the cornea will be sterile, and you then should enter a healing phase. If not, then it's progressive keratitis. So we're going to continue work on the bacterial ocular surveillance system with our Australian Vision Research support, and this will give us trends on the national distribution of microbes and their resistance pattern in keratitis. The centres, this is a list of the centres that will be included, and we're currently collecting data from them. And once we have the system set up, this will be self-sustaining. So the funding is to set up the protocols and coordinate the system so that this in future years will be able to be performed without the need for as much resources. So just to finish, keratitis is simple and complex. We need epidemiology to inform our drug choice, and that's critical, and this is where the BOSS fits in. Once we have the BOSS data, we can then look at our existing guidelines and make sure they align with our data. But we still need to advocate. We need to advocate for microbial keratitis to be considered a neglected tropical disease, to get the resources behind it to cure more blindness. All national centres are welcome to join. Please reach out if you would like to be part. And I'd like to make a special call out to Dr. Maria Carrera-Argas, who's really worked this up, and the other co-investigators on the grant, which are listed there. Thank you, and thank you to Australian Vision Research.
Thank you, Stephanie. I'd like to open up the floor for any questions. I don't know if anyone's put in questions through the app.
While the questions are coming, I'll just remind everyone that on the program there's little stars, Australian Vision Research stars, and that's where the Australian Vision Research Board has looked at the abstracts and sort of pinpointed the ones that we've found to be of interest. Yeah.
Stephanie, can I ask you a question?
Yeah.
I mean, it's very interesting to look at the lab data about the resistance, but I was wondering if you had any clinical data on the use of a bacteriostatic and a bactericidal antibiotic, adding chloro to one of the other antibiotics, because the teaching is that you should use, not use a cidal and a static antibiotic together. So how's that worked out clinically?
Yeah, we have 5 years of clinical data on microbial keratitis that we've published the outcomes for, and that in that data we have compared the clinical outcomes from using chloramphenicol with fluoroquinolone versus fortified therapy, and the clinical outcomes were similar in terms of effectivity, but the fortified antibiotics had a higher risk of having complications. So the clinical data, and we've been working with the microbiology team. I mean, when we give topical antibiotics, we give them in very, very high doses, extremely high. The microbiologists tell me it's like, you know, having a sort of a wheelbarrow full of antibiotics and sort of chucking them at the eye. So clinically, we've noticed that it's similar.
Obviously, the fortified antibiotics are one thing, but if you look at, you know, Cipro or ofloxacin with or without chloro.
No, you need the chloramphenicol to give you the greater gram-positive cover, because the fluoroquinolones don't provide enough gram-positive cover.
I've got a question up the top. Jeremy Rater from Perth, Western Australia. I was also interested in the bactericidal and bacteriostatic approach Professor Hugh has said, because it was classic teaching that you couldn't combine the two. They had very little effect. That was one comment. The other comment was that most of the results that we get from the microbiologist is that the chloramphenicol is resistant, that there's resistance. And in fact, when you talk to them, as you said, that the percentages that we use is much higher than what they would normally do. So what do you suggest, what can we suggest to the microbiologists when they recommend that chloramycetin is not the appropriate drug because it's resistant?
Yeah, so it depends. Like, some labs will report chloramphenicol sensitivity testing on gram-negatives when we know it's going to be resistant anyway. And this is why we need to get sort of more national data, because our experience is that the cefalothin resistance is higher. The other issue is that, these the MICs and resistance data are based on systemic breakpoints, and so it's still being determined. They're not ideal in terms of determining ocular patterns of drug response, but when we've looked at our clinical data, cases that have resistance to the antimicrobials that are on actually do have poorer outcomes for some antimicrobials. This is a project we're working on with experts in microbiology and running surveillance systems. Professor Monica Lara, she runs surveillance systems for other microbes across Australia. And there is more we need to know. I'm also part of the MOG group in the UK where we're looking to see, was there a better way to actually test and determine what is the best antimicrobial. The data I've presented is our surveillance data, and we've done some equal clinical studies. But yeah, the work continues. Thank you.
If there are no other questions, I think we'll wrap up as we're already eating into your lunchtime. So thank you everybody for staying, and as RANZCO Fellows, you're all AVR members, so thank you for your ongoing support of Australian Vision Research. You can see the important work that we're doing. Thank you.
Thank you.